Hello everyone, myself Sakshar Shah. I'm an assistant professor from the Department of Pharmaceutical Technology, JS University. Today the topic I'm going to teach you, that is bioassay, is a part of the subject pharmacology 2. It's a subject of B.Pharm Sec, third year, with the subject code BPT 503. So today we'll be talking about what is the importance, what is the definition, principles, and what are the different types of bioassay we can have. So let's see what we have for today. So biomass is defined as the estimation of the potency of an active principle in an unit quantity of preparation, or detection and measurement of the concentration of the substance in a preparation using the biological methods. So basically it's a detection. So you know if you divide this total word bioassay, so it is coming bio and assay, and bio means biological, assay means itself an word assay. So assay is basically meaning whenever you're going for chemical assay, so it's becoming the method of doing the percentage, calculating the percentage purity. Like if a substance we are considering how much purity, that means how much real substance is present in that particular material. So that is the percentage purity calculation. And bioassay means biological percentage purity, means in a biological fluid we want to detect or measure that how much amount of the particular substance is present in your body. That could be any biological elements like any hormones like oxytocin, vasopressin, or if you're taking any drug, so how much amount of drug is basically present in your body. So that test, okay, that method or that phenomenon can be called as bioassay. Importance of bioassay is that sometimes some of the detection or measurement cannot be accomplished by the chemical or physical assay. That time bioassay comes in the place. Active principle of the drug is unknown or cannot be isolated, like insulin, posterior pituitary extract, etc. Chemical method is either not available, or if available it is too complex and insensitive or requires higher dose, that is insulin acetylcholine. Chemical composition is not known, and long-acting thyroid stimulants. Chemical composition of drug differs but have the same pharmacological action and vice versa. That is cardiac glycoside, catecholamines, etc. Principles of bioassay. The principle of bioassay is very easy, and you need to understand it. That the principle is basically a theory of comparison. Comparison between the fact you know and the comparison between the fact you know and and the fact you want to know. That means for a particular substance, if you want to know the particular action, like suppose you are having a test drug which you are assuming that could have a uh, gastric gastric acid secretory property. Okay, and now to know actually it is showing some response, what you need to do, you need to collect a standard agent from the market which is having the gastric acid secretory property. So the both you will be injecting to different group of animal, or in vitro or in vivo study, and then you will see what is the response, and you will be comparing between the response of the standard one and test one, and the result you will be getting. So from there you can get some conclusion whether your test substance is having some action which you are assuming or not. So this is basically the comparative study, and I'm sure you have must have heard about it. I have already discussed in the previous semester laboratory that the concentration of test, concentration of standard, we have to take a group for blank, or so and so. So this is basically the principle of bioassay. Next is types of bioassay. Basically, two types of bioassay is there: quantal bioassay and graded bioassay. So we need to know what are those. Quantal bioassay is the response is in the form of all or none. Either there will be response, there will be no response, or there will be maximum response. There will be nothing in between. And drugs producing quantal effect can be bioassayed. That what is the effect by the endpoint method. What is endpoint method? We'll discuss later. Next is graded bioassay, or graded response is the response of proportional dose. Like The previous one, the quantile assay, we have talked about that it is the the some value at the end point or at the edge. Like either it will be having it will be having no action, or it will be having maximum action. There will be no something in between. But what is the part of between? So the assay which calculates the doses which lies in between that two ultimate dose. So those assay is being called as a graded assay. The different types of methods are being can be taken place by which we can do the graded assay. First, bracketing or direct matching, interpolation, multiple point assay. In that, 3-point assay, 4-point assay, 6-point assay is there, and cumulative dose response is also there. So now methods for a bioassay of agonist. So we'll be talking about a endpoint method. So agonist, the reason we have mentioned here agonist, because this agent will increase some action or potentiate the basic action. So that's why it will be considered as agonist. And we'll be talking about here the digitalist. I'm sure you know the cardiac alkaloid digitalist, you know. So the threshold dose producing the predetermined effect is measured. Threshold dose means the dose at which it shows some response. Comparison between the result of test and standard is being done, and we'll be talking about the bioassay of digitalist in cats. So the cats are anesthetized with Fluoralose is an anesthetizing agent. Its blood pressure is recorded. The drug is slowly infused into the animal. So the drug is given slowly, and the blood pressure. The blood pressure is continuously being measured. When the blood pressure falls to zero, there is no blood pressure. The volume of the fluid infused is noted down. How much fluid you have given. Two series of such experiments, one using Digitalis, another using your test preparation is being done. Now potency are being calculated. So potency can be calculated by concentration of an unknown is the basically the potency. So is equal to threshold dose of standard by threshold dose of test into concentration of standard. Next, matching and bracketing method. A constant dose of standard is bracketed by varying dose of test sample until an exact matching between the response of standard and that of the sample is achieved. Initially, two responses of the standard are taken. The doses are adjusted such that one is giving response of approximately 20% and other 70% of the maximum. The response of unknown, which lies between the two response of standard dose, is being taken. So here the standard two dose of standard are being taken. The panel is repeated by increasing or decreasing the doses of the standard till all three equal responses are obtained. The dose of test sample is kept constant. At the end point, a response of double dose of standard and test match each other are taken. This should give an equal response. Concentration of the test sample can be determined by concentration of unknown by dose of standard by dose of test into concentration of standard. So here you can see some bioassay graph of bioassay of histamine on guinea pig ileum that we have got, and this you have done also in the you can T and SMIN test and standard, and you have seen this in the software in the laboratory in the last semester, I'm sure. Now the graphical method. This method is based on the assumption of dose-response relationship. Long dose response curve is plotted and the dose of standard producing the same response as produced by the test sample is directly read from the graph. In the simpler design, 5-6 responses of the graded dose of standard are taken and then two equative response of the test sample are taken. The height of concentration is measured and plotted against the log dose. So the characteristic of log dose response curve is that it is linear in the middle. Thus the comparison should be done within this range only. In the other words, the response of the test sample must lie within this range. So these are few methods by which you can do either graded or quantile assay. The rest part we'll be discussing in the next class, but this is something completely new to you, but The thing what you need to do is understand it. As pharmacology, the subject that you cannot mug it up only, you cannot keep in mind, you have to understand, then you have to keep in mind. So if there is any questions you have, you did not understand, I'll request all of you, please go through my slides and hear what I said. But still not, if you did not understand, please come to me in the next class, and I'm sure I'll discuss about it in the next class or doubt clearing classes. Thank you so much.